Hello, we are back. And I didn't check my mic earlier. Hopefully it now works. But yeah, so today we are going to discuss amino acid metabolism. For my master's, well, this might be doxing. But I do have a master's. Okay, wink, wink. So let's go with amino acids. It serves as both building blocks for patients and versatile metabolic intermediates. And while proteins are their primary function, amino acids are constantly being turned over, and proteins are broken down and rebuilt, and their carbon skeletons can be used for energy or biosynthetic precursors. And as we know, around 70% of amino acids are recycled during protein turnover, and that would be like for the highly dynamic amino acid pool that interacts with many metabolic pathways. Okay, so if you think about it, so where we get the amino acids, it's from proteins, the body protein, your diet protein, and the synthesis of non-essential amino acids. And after they become like the mush of amino acids in your body, they become your body protein and would help synthesize different things such as porphyrins, creatinine, neurotransmitters, purines, pyrimidines, and other nitrogen-containing compounds. Okay. And then, with that, one major concern actually is ammonia, and it's a toxic byproduct of amino acid degradation. And the body handles this by converting ammonia to urea in the liver, and it is then excreted by the kidneys, and this detoxification process is vital for maintaining metabolic homeostasis. Okay, so just remember, ammonia is toxic, we have to remove it. And amino acids are not essential. Wait, actually, let's try if this works. Will it work if I draw it? So yeah, where is it? Okay, it's here. There you go. So yeah, here. It works! It'll be horrible because I'm not used to writing using this specific tablet, but it's alright. Dietary protein, and you have an arrow here, and then you have the body protein. Wow, this is funny. Okay, and then you have the synthesis of non-essential amino acids, and all of those becomes your like mini amino acid pool, swimming pool. And then after that, they become again your body protein, and will like those will be used to synthesize like neurotransmitters, purines, and pyrimidines. Okay, yes, that is a artistic interpretation. Alright, next one is most amino acids are not essential, and they can be made by interconverting other amino acids or from other metabolites, and essential amino acids must be obtained from the diet. And you have non-essential amino acids such as the alanine, arginine, asparaginase, aspartate, cysteine, glutamate, glutamine, glycine, proline, serine, tyrosine. And your essential ones would be PVT, timhol, that is phenylalanine, valine, threonine, so PVT. Oh, that's a double T, so PVT, timhol, threonine, tyrosine, tryptophan, isoleucine, methionine, and then histidine, arginine, leucine, lysine. So again, the essential amino acids are PVT, timhol, phenylalanine, valine, threonine, tryptophan, PVTT, isoleucine, methionine, histidine, arginine, lysine, leucine. Okay, good. Again, one more time. The essential amino acids are PVT, thymol, phenylalanine, valine, threonine, and then tryptophan. Why do I always say tyrosine? No. Again, phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, lysine. Okay. And then okay. When not needed for biosynthesis, they are diverted to catabolic fates. So their nitrogen is removed, and no, that's alright. So the nitrogen is removed, and often producing toxic ammonia, and that is later excreted as urea, and the remaining carbon skeleton, aka the part without nitrogen, it enters the central metabolism. So, and with that, the remaining carbon skeleton, the part without nitrogen, it enters central metabolism. So that's how you just recycle them. And after you break it down, the carbon backbones will be used to make, voila, which one? Glucose, another thing that's very important. So glucose would be the glucogenic pathway, ketone bodies would be the ketogenic pathway, fatty acids, or even it's also fatty acids can be made, obviously. And then you can also just directly oxidize them to CO2 via the TCA cycle. Funny thing is that it has 3 names, TCA and Krebs. What was the other one? Because TCA is already the tricarboxylic cycle, I think. And I mean, okay. Now, for amino acid oxidation, oxidases will do that. So usually, the oxidases are enzymes, which has the cofactors, okay? They have FAD or FMN, and those would deaminate your amino acids. So for example, if you have an L amino acid, and if you add an L amino acid, you make it into an alpha keto acid, right? And in the process, you release an ammonia, and that would form the hydrogen peroxide. So in the first step, you essentially have L amino acid and water, and then you have the alpha keto acid and H2O2. So that's usually the first step. And another one example is L-glutamate. Yeah, this one's a specific one. So you have L-glutamate and water, and then if you exchange it with the glutamate dehydrogenase, oxidation will happen, and then you have alpha ketoglutarate plus ammonium. So ammonium NH4 plus, okay? And NH4 plus. Hello, welcome Light Neon as well, and David too. We are now on the glutamate as the central nitrogen hub. So most amino acids do not directly release their nitrogen quickly, and instead they will transfer their amino group to alpha-ketoglutarate through the different trans-amino reactions, and that would produce glutamate and corresponding amino acid. So we talked about that one. How can I erase the board? I want to try it. Grabbing the pencil. Okay, now it's this one. Where's my eraser? Nice. Okay, good. So I'm gonna erase this, and then if I just need to write it out, There you go. So you have L-glutamate. My handwriting is usually normally prettier. It's just I'm not used to this tablet, okay. H2 plus water. And then it becomes alpha ketoglutarate plus NH4 plus. Beautiful. Hopefully that's clear enough. Oh my gosh, it's too... We need to make the pencil a bit thicker. Okay, one more try. So I'll write it this way. You have L-glutamate. Does that work? Okay, nice. Plus H2O. That one will lead to alpha ketoglutarate plus NH4 plus ammonium. Nice. It works, you know, technology. Okay. Then we have the interconversion of amino acids, or using like transaminases. So transamination reactions allow nitrogen flow between amino acids, and most of them can be transaminated. And you actually would use PLP or pyridoxal phosphates as a coenzyme, which is derived from vitamin B6. So we have two key reactions that I could attempt to draw here with our artistic interpretation of how it should look like. Oh, wait, this one. Is this the pencil? Yes. Okay. Work. And here. Yeah. Hopefully this one. Okay, that one works. So you have L-glutamate, right? Glutamate plus pyruvate. And then you have the ALT or it could be glutamate pyruvate transaminase. I like calling it GPT because it makes sense, glutamate pyruvate transaminase, but unfortunately it has a different name as well, called ALT, alanine transferase, and that would lead to alpha ketoglutarate and alanine. So yeah, it depends on which one you want to call the enzyme, but yeah, you have L-glutamate plus pyruvate, and then if you have the glutamate phosphate transaminase, then you have the alpha ketoglutarate and L-alanine as a result. And then you also have another different one, would be L-glutamate plus oxaloacetate, and same one, you can use GPT or ALT, and it's still alpha ketoglutarate and L-aspartate as a result. So it just depends on the amino acid you're using, if it's like the L-glutamate, if it's like from pyruvate or oxaloacetate, because they're both coming from L-glutamate. And take note, I think I mentioned this before, transaminases rely on PLPs, which forms a Schiff base with the amino acid substrate. Okay. Oh, we talked about electron negativity before, but the basic idea would be two types of chemical bonds. They are ionic and covalent. But the thing is, different atoms have different interactions with different electron orbitals. Did I say different 3 times in one sentence? Yes, I did. So just to make it more confusing. And then what do we expect? Example, you have NaCl. That's a very, very classic ionic bond, and that may have like around 25% ionic character. I mean, like, it's not 100% ionic. It kind of has some covalent characters as well, but just remember those. It's kind of a mixture. What are the factors that would influence bond polarity? So there's electron affinity. go. Let me just erase this masterpiece here. There you go. Okay, that works. So you have two things. You have electron affinity, and then the second one is ionization energy. I will be using shortcuts to make it faster. So electron affinity is how strongly an atom attracts electrons, and then ionization energy would be how easily an atom loses the electrons. Again, electron affinity, it's like how strong its pull is, how strongly an atom will attract electrons. Ionization energy is how easily it can let go to lose electrons. And both of them would contribute to another thing that we always hear: electronegativity. And in the periodic table, electronegativity increases across a period, which is left to right, and then that's because of its increasing nuclear charge, but it decreases down a group because outer electrons are farther from the nucleus and they experience more shielding. And that's where you get the terms called bond dipoles and molecular polarity. And you also have the reactivity patterns in organic and inorganic molecules. And that's also why, hello, Zoro is always lost 247. Oh my goodness, I can't believe Zoro is also here in the stream. Yes, hello X-Job. Yup, note those down. And that's also actually why a lot of these things just coalesce or come together, and that's why you have the bond dipoles, molecular polarity, reactivity patterns in organic and inorganic molecules, and that's also why oxygen, nitrogen, and fluorine would dominate the hydrogen bonding in biology. You always hear them. Those are common, the very common ones. Okay. And we talked about the electronegativity. Oh, intermolecular interactions. Let's try that. So let me erase this first. So for intermolecular interactions, there are some common terms also that you end up hearing always. First is one. You have hydrogen bonding, and that's when water would form like a structured network. That's a special type of dipole interaction, which I will also write here. Dipole. And so in dipole you have polar molecules like HCl which will align, so they have partial positives. Oh, the little cute characters are all bouncing on the screen. That's so nice. Yeah, so polar molecules like HCl would align, so you have partial positives and negatives that attract, and that would form the dipole attractions, which stabilize molecular assemblies. And you also have van der Waals. Yes, that's not it. 3 van der. I think this is how it's spelled. You have van der Waals or London dispersion forces. Yep. So the weakest one here would be van der Waals, because those are just like non-polar molecules interacting via the instantaneous short-term dipoles caused by their random electron movement, and these can induce dipoles in the neighboring atoms, creating temporary attractions actually. So that's why this phenomenon would also contribute to uncertainty in electron position. And then we have a thing called the the hydrophobic effect, which is not a part of these groups. That's, or more on, they're different. It's not the London dispersion force. Let's go with this brush. So the hydrophobic effect would be focused on water. Okay, so it's not due to a specific bonding interaction like the van der Waals forces. That instead it would be because of the entropic force. Oh, it brings back lectures in your biochem. Welcome back. It will haunt you. It's still here. It doesn't end. Whether it's in 9th grade or higher, it's always there. Yeah. So entropic cost of organizing water molecules around the nonpolar substances. So here's the thing. When a nonpolar molecule is introduced into water, water is polar, right? So the polar water molecules would rearrange to form like a cage structure around it. And this structuring is not favorable. And as a result, the non-molecules the non-polar ones would just stay inside each other. So I'm just gonna try and you know the cell membrane thingy where you have the tails and the head thing? So the tail, those are non-polars, and that's why they try to stay in the middle away from the water. Oh my gosh, wait, I think I can choose a different color. Look at that, artistic. Yes, so if the water is around here, the polar heads would be the ones around near the water, while the tails, the non-polar ones, would just stay inside. Amazing. Oh, and then next is like the cis-trans isomerism in alkenes. So and organic chemistry is still here. Okay, let me just erase this. Okay, there you go. Almost there. Sorry. Okay. Not the blue one. Okay, so you have cis butane 2-ene. What are those? So you have, this is alkene, right? And then this is methyl. That's why it's cis, but what's the but? Because of the 4 carbons, C, 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 C. And then it's cis because it's like in one side. Meanwhile, the trans butene would be like this, or of course like this. Beautiful. So opposite sides. Which one would be more natural, or like what position do they like to stay in? It's gonna be in the trans isomer, because the opposite sides would lead to like less steric hindrance, and that would also lower the energy. And so you have the pi bonds and all that, but we can just go through that in a future one maybe. Okay, so we have some reactions. You have the nucleophilic addition to a carbonyl group. So let's just write the general reaction here so that we have a guideline here. The general, yes, thank you. The biochemistry is nice. You just have to like dig deep and do soul searching, and then you end up like liking it too. Okay, let's write. This is R C O H. So R is just like any methyl or something that will go there. But if an XH attacks something plus H, you have addition, and then this becomes alcohol. You have R, and then X is also here, and okay, I could just write the H. So that's addition one. And then you could have elimination, and then like water would leave, so you have H2O. And what's left? You have R, C, double bond to X, and then H. So that's how you go from the carbonyl. Oh no, can I back it? Yes, technology. From carbonyl to alcohol, and now you have a C double bond X, and then I think there's a fourth one. If you go here, if it fits, you have C, X, X, H, R. Beautiful. So now we have a CX, the carbon and something. So that's how you just try your best and make it different nucleophiles. Okay, now you. Oh yeah, we talked about the isomerization of the different bonds. So just mention this. Okay, so in the transamination mechanism, there is a step which involves the isomerization of the imine double bond followed by hydrolysis, and you have the shift base immediately rearranging, and that would shift the position of the double bond, allowing the addition of the water, which shows there, and this breaks the bond between the amino acid and the PLP. Okay. Okay, now let's try and we mentioned the PLP numerous times, but what is it and why is it important? So I will try to draw it here. Okay. Oh no, this one. So the PLP is, you start this, will it fit? C, O, oxygen. Oh, that's a hydrogen. Okay, this is a like that bond, dot. It's temporary. And then C, H, R, and C, carbon, O minus, and okay, then you have another one here, N, H, C, H, 3, and a line, a bond, another bond, carbon, CH2, I didn't need to write it, O, P, O, H, double bond, O, now we did it. So this bond will be labelized by the transaminases, and this one will be affected by the decarbolases, and then aldolases would touch this one. So remember, the decarboxylases would cleave the bond between the carbonyl and alpha carbon group. So this one, decarbolases, transaminases would be at the right, the alpha carbon and carboxyl group. No. Alpha carbon and hydrogen. And then aldolases would keep the bond between this one, alpha carbon and the R group, the side chain. Nice. So it has a very electron withdrawing nature and a conjugated ring system. So that's why it's really used as intermediates during the reaction. Okay, and then now we have the urea cycle actually. So the urea cycle, that's coming from, you have the glutamate to glutamine, right? So if you want to start, glutamine synthesis catalyzes the ATP dependent synthesis of glutamine from glutamate. Okay, actually before yapping, I'll just write it here. Let's Ohio. Hello, Georgie. Well, George, welcome back. We are at the urea cycle, so let us scribble it down. We have new technology, by the way. I apologize for my horrendous handwriting here. It's not usually ugly. It's supposed to be pretty. I'm just not used to using this one. So you have glutamate, and then you have glutamate synthase. ATP dependent, so you have ATP into GTP, ADP. Oh my gosh, do not mix those up. And then you have an intermediate over here, and then another this GS, but this time it's ammonia into PI, and then you have glutamine here. Beautiful. Okay, so glutamate to glutamine. If you want the specifics, you have glutamate synthetase catalyzing the ATP dependent synthesis of glutamine from glutamate and free ammonia. Again, if you want to circle those, glutamate synthase catalyzes the ATP dependent synthesis of glutamate into glutamine. Synthesis of glutamine from glutamate to free ammonia. Lamau. I think at some point I'll talk about, maybe, I don't know. Google. No, don't Google. Go to a doctor if you really have kidney stones, please. Do not die early. Water. Make sure to drink water. It's the solution to a lot of things. Okay, then we have this one. Ammonia is transported. Oh, now we're talking about where it goes. So ammonia is transported to the liver, where it is ultimately excreted as urea. And the immediate precursor of urea is the amino acid arginine. And then within arginine, the guanidino group would contain the structure that forms the urea molecule. And arginase would cleave the arginine to release urea and ornithine. What did I say? Okay, I'm a visual person, so I have to draw it out. Here. If you can hear the erasures, that's me. Okay, no, not red. Kuro. You have arginine, and then you have urea and ornithine. Okay, so again it mentions that within arginine it has the guanidino group that forms the urea molecule. And what does this? Who is at fault? Arginase is the magic syrup that helps that happen. So ammonia is generated from amino acid metabolism, okay, and it's primarily cleared by the liver again through its conversion to urea for excretion. So this one, you don't like that. It has to be, you have to get rid of it. And once in the liver, glutaminase hydrolyzes glutamine back to glutamate and free ammonia, and then it enters the urea cycle. Okay. Erase next to you have the urea cycle again. In the urea cycle within hepatocytes, the rate determining step is the synthesis of carbamoyl phosphate from ammonia and carbon dioxide. New characters. So many new characters. Wait. Okay, there. So you have, imagine this is the hepatocyte. So you have ammonium and H4 plus, right? And you have carbamoyl phosphate, and then you have ornithine, and then so the ornithine would become citrulline here outside. and there's also ornithine here. I'm gonna talk about it in a bit. Okay, again. So the rate determining cycle is carbamoyl phosphate from ammonia and carbon dioxide. Oh, I did not draw the carbon dioxide. CO2 was here. And it happens in the mitochondria. Right. This is supposed to be green. It happens in the mitochondria and requires ATP. Mitochondria is what? The powerhouse of the cell. Mitochondria is the powerhouse of the cell. Okay. Then carbamoyl phosphate then reacts with ornithine, and it is transported into mitochondria to form citrulline. And the second reaction is catalyzed by ornithine. Oh wait, the carbon reacts transported into the mitochondria to form certain. No, this isn't. Yes, it is classic. Now this one would go to the mitochondria later on. There you go. So it's after the citrulline would be there in the mitochondria area. And then the enzyme arginosuccinate synthase catalyzes the reaction between citrulline and aspartate using ATP to form arginosuccinate. So again, the urea cycle is within hepatocytes. Change the color. This is pink. It's within hepatocytes. Okay? Hepatocytes. Hepatocytes. And later on it just gets delivered to the TCA once it has citrulline already. Yeah, there's a bigger figure, and as you can see, I'll just scribble it here. Citrulline, and then arginosuccinate synthase, this one. And then you have arginosuccinate, you have fumarate, which is also a very familiar thing. Arginine, again, we talked about that earlier. Arginine, if you have arginase, becomes ornithine. And then the thing is, from arginine you also have urea, which is excreted to the hepatic vein. So it's a rough drawing, but do you see what's happening? The pink one is this is all happening inside a hepatocyte. Okay, they're just labeling them with a different color now. And so you have glucose, right? Yeah, that if you start from glycolysis, you have glucose to G6P, blah blah blah, and back. But if you look at it from a different perspective, they all talk to each other. So that's why it's important. Like the glycolysis, gluconeogenesis, oxaloacetate from TCA, succinyl-CoA, fats and fatty acid, they all talk. So if you just want to piece them all together in a very simplified metabolism flowchart, it goes like this. Where is it? Where's the pen? Okay. I think I should make this one a tad bit smaller. There you go. So we are familiar with the TCA cycle, right? So you have oxaloacetate, citrate, and you have citrate. Isocitrate, alpha ketoglutarate, succinate, fumarate, malate, and then there's something, and then oxaloacetate. But oxaloacetate can be converted to aspartate, which goes where? To the urea cycle. Is aspartate, and then you have the ASA over here. Arginine, you get rid of it as urea later on, becomes ornithine, and up here is the citrulline. And your CPS, remember the CPS from the mini picture earlier, carbomonophosphate from NH3 plus HCO3 minus. Yes, I think we're missing something. Glutamate also comes in here. Somewhere here. Where does glutamate come from? Aspartate. Alanine. Alanine. And alanine also makes pyruvate. Which makes what? Acetyl-CoA. And where does acetyl-CoA go? Here. So the arrows are all connected, okay? The arrows are all connected. And then, yeah, the intermediates are all shared via transamination. And they are all trying to donate nitrogen to the urea cycle. And in return, the urea cycle would, I forgot to write the crucial part. They would all donate nitrogen to the urea cycle, while the urea cycle would give them fumarate, which enters the urea cycle here and helps regenerate oxaloacetate. You have fumarate and malate. Fumarate. Artistic interpretation. Yes, doctor's handwriting. Thank you. I try my best to make it as pretty as possible. It's not working out. It's normally not this horrible. Okay. As mentioned before, the glucose-alanine cycle is a nitrogen disposal pathway that links glycolysis in the muscle with gluconeogenesis in the liver. And in the muscle, the pyruvate generated from glycolysis would accept an amino group from glutamate via alanine transaminase, or ALT, and you form your alanine. Alanine travels through the blood to the liver, donates amino group to alpha-ketoglutarate, you regenerate the glutamate, and then you release the ammonia for urea synthesis. Essentially, you just want to get rid of urea. And so the organs that we talked about were the intestine. Intestine uses glutamate as fuel and releases alanine and citrulline into the circulation. Alanine, this one, citrulline and alanine. I don't know if oh my gosh, you could see the cursor. Beautiful. The liver would receive amino acids from the intestine and the muscle, especially alanine and branched chain amino acids. And then you have it also performs gluconeogenesis, producing the glucose, and urea synthesis for nitrogen disposal. Again, so what's intestine for? It uses glutamine, and it uses glutamine as fuel and releases alanine and citrulline. Glutamine as fuel and releases alanine and citrulline. G into AC. And then you have liver. It receives amino acids from the intestine and the muscles, especially alanine and the branched chain stuff. The liver would perform gluconeogenesis to make glucose again. It also does urea synthesis for nitrogen disposal, and it would send glucose back to the muscle. It also releases citrulline. The muscle would then take up glucose and BCAAs and release the alanine and glutamine. Alanine would carry the nitrogen to the liver. Okay, I think it would. Let's just try and piece them all together over here, just to make it look easier. And where's my marker? Hello, Landor. Welcome back. We are trying to do the inter-organ relationships in amino acid metabolism. So again, you have, where's the intestine? Intestine would look kind of like this long stuff. So it uses glutamine as fuel, and it would release alanine and citrulline into the circulation. And the liver, whoa, how does it? Nice. And then you have all amino acids going to the liver there and here. Okay, the liver receives amino acids from the intestine and also the muscle. So now you have the muscle over here. Muscle. You have alanine from the muscle and branched and also especially alanine and branched chain amino acids. I think you have BCAA. And then it performs gluconeogenesis, urea synthesis. So glucose, you make glucose, you also make urea. And then after that, it also releases citrulline. Which goes where? Citrulline goes to the... This is a kidney. Okay, citrulline goes there. You also have the glutamine from the muscle going here. So citrulline and glutamine going back to the kidney. And it is secreted as arginine, NH4, converts it there, releases ammonia for pH regulation, and glucose for energy. Look at that. It's all connected now. Nice. And yeah, I think that might be a good stopping point because the next one is a bit different now. It's digging more into ammonia and all that. Yes, thank you so much for joining me here today. I hope my handwriting was legible here. And yes, that is all for now. The karaoke, don't worry. So far I'm trying to cut the ones that could be released. I think I'm just trying to avoid the Disney ones because we did the what was that called, High School Musical, Hannah Montana, and a couple more, but the rest that's not Disney I will clip them. Okay, so slowly but surely they will be released. I think one mini clip was already released. It was the Lupang Hinirang, the Philippine national anthem. So yeah, but thank you for watching, and that is all. Bye bye.